So yeah, I'm JB. I work in the autopilot lab at University of Chicago, and I'll talk today about bioinformatic pipelines that I've developed with Parcel. So it's gonna be a little more bio-heavy, sorry, but yeah. It's important to note, first of all, that the tumor is not only composed of tumor cells, right? A lot of immune cells can actually infiltrate the tumor, and so there's this new group of therapies called immunotherapy, which are based on the idea that you can use these immune cells to fight cancer. And so it's very important to understand this very complex tumor-immune relationship. There's lots of variability in this relationship. So some tumors have very little inflammation, whereas others have lots of immune infiltration. So cold versus hot tumors, but also the role of immune cells can vary a lot. Well, typically the immune system is supposed to kill any abnormal cell, but in some cancers, the tumor is actually able to modulate the environment and escape immune surveillance. And so in the lab, I'm mostly focused on this relationship of a tumor cell presenting an antigen to the immune system. But today we're going to focus mostly on this specific area, so T cell receptors and B cell receptors. As you can see, there's lots of potential diversity in terms of T cell receptors, and the same is applicable also for B cell receptors. And so this diversity is reached through VDJ recombination. I'm not going to go through this in too much detail, but basically you have kind of 4 sets of genes, V, J, C, and D. And so you can choose one gene from each group and also have a lot of deletions and insertions within this rearrangement. And that's how you create very specific receptors to target an antigen very specifically. So that's how you got kind of the specificity of the immune system, in this case for cancer cells. Our pipeline is essentially a consensus of multiple algorithms which extract T cell receptors and B cell receptors from RNA sequencing. And so the general idea, well, when you start off, you have bulk RNA sequencing, so unselected sequences. And so you have very low percentage of reads that are actually mapped or are actually like related to B cell receptors and T cell receptors, as you can see here in one. What you do, first of all, select the sequences that are mapped either to those VDJC genes or sequences that are unmapped. And then you can remap them and recombine them and recreate these sequences and extend them using reference databases to kind of get this final output of the TCR and BCR of a specific sample. When I first started out this analysis of B cell and T cell receptors within our samples, I was only working with Trust3, this algorithm here. And so I noticed that there were a lot of issues with the outputs. So there are a lot of fragments, there were duplicates and also non-canonical sequences. And so I decided to try out different algorithms, and that's kind of how I came up with this whole pipeline. And so, yeah, basically we run multiple TCR and BCR extraction algorithms and also all their dependencies, and then do different filtering steps to remove non-canonical sequences and such, and then merge all the results of these algorithms and kind of come up with this consensus call at the end to kind of have this final output. Just briefly, there's a lot of downstream analyses possible from these results of our pipeline. So the first most basic one is just kind of estimating the B cell and T cell infiltration within the tumor. You can kind of see here like the graphs of different diversities, the diversity of the TCR alpha receptors within one of our samples, for example. But also kind of more downstream, you could also look at the relationship of these TCRs and BCRs with antigens. So there are different tools to cluster BCR and TCRs into groups that are predicted to bind to the same antigen. We can also look at public databases about this kind of analysis. And also one really interesting thing would be looking at spatial temporal analysis. So looking at different regions of the tumor and looking at how the TCR and BCR repertoires kind of overlap. But also you can do that throughout time, for example, like a patient, and look at over the course of a treatment, how the TCRs and BCRs evolve within that individual. Now to the Parcel part. So all the pipeline elements were parallelized with Parcel. So the bioinformatic programs that as you would expect, are all Bash apps, and then the filtering and merging steps are done through the Python apps. And so we kind of have this one command line to automate the whole process, which makes it very useful. And also it allows a lot of modularity. So these are only the kind of basic parameters, but you can kind of configure it a lot more. And so obviously there's a lot of advantages, specifically like speed up, obviously with parallelization with Parcel. But also we've implemented Docker and Singularity as part of this pipeline. So this allows a lot of reproducibility. Anna, who's also on the Parcel team, is kind of our chief reproducibility officer. And so she's very much pushed for this because that's kind of an issue in bioinformatics. And so, but this also makes it a lot more easy to deploy using Docker and Singularity. And then finally, but also probably most importantly, this can also improve sensitivity or specificity, sorry, of this analysis. So depending on what quorum you choose at the end, you can kind of either capture more TCRs or kind of really select only TCRs and BCRs that are known to be true. And so Anna kind of told me to talk about things that are that could be improved with Parcel. So I thought I'd first start with everything that I really liked about Parcel. So I really like the fact that Python and Bash scripts are really easily integrated. That makes it very easy. And also, I really like the modularity for executor creation. So specifically my pipeline, for example, some of the tools are require a lot more resources than others, like alignment, for example, requires 40 gigabytes of memory. And so I'm allowing the user to kind of allocate resources a lot more specifically for each step. And also I've tested this pipeline on on Torque and Slurm, but it seems very portable, so that's great. So potential improvements, I've been a little frustrated sometimes because when you kill a Parcel command, it doesn't automatically kill the jobs that were created by this command. And so that can sometimes be a little bit of a pain to kind of manually kill those commands. And then another small possible improvement would be allocating executors at runtime. So right now, since I'm allowing the users to specify which configuration, but also which tool should be allocated to which executor, I think it'd be great to have this be done at runtime too. That's just a small idea. Anyways, thank you everyone to Parcel for implementing this tool. It's very useful. I also want to thank everyone in my lab for help in my research.
Wonderful, thank you, JB. A photo from the times when you were actually in person, looks very nice. Any questions? I guess we're a couple of minutes late, but I think we got time for a question or two. I personally am intrigued by the container stuff, but I will wait to see if others have questions first. Okay, I will go ahead with my question then. So Chief Reproducibility Officer Anna made you use Docker and Singularity containers. So how are you integrating that? Are you basically packaging each of your tools, or do you have one Uber container? And then how are you managing the difference between Docker when you're running on wherever Docker's running and Singularity when you're running on wherever Singularity is running?
Yeah, so the user is able to specify whether they want to run locally with Docker or Singularity, right? And then as to the Docker files, so some of these tools already have Docker implemented, so I'm not actually I'm just using their provided Docker files, but I also did create the Docker files for specific tools. And so basically what happens is that you have, within each function, depending on what the user specified, I can either wrap it with a Docker container or specify the local installations that the user provided, or run the Singularity using the Docker file that was created for each algorithm.
That's very cool.